Namaskaram. In this session, we are discussing the secondary growth in dipole root. So, in plants, for the angiosperms or gymnosperms or almost all the plants which show the presence of root development and the early stage or in the primary growth, and the establishment of the root system is usually taking place during the secondary growth. And as part of the secondary beginning in stem, in dicotyledonous plant, the root may also show the secondary beginning. So here, what we have to understand that in monocotyledonous plants, there is no secondary growth or secondary thickening is taking place in root system. Only in dicotyledonous plants. Okay. So normally it is present in most dipole roots in plants, and what we can see normally in the vascular system, the secondary growth which is organized or developed by the cambial activity. So, as in the case of root system, there is no vascular bundle-like formation in the ground tissue. Only the radial arrangement of xylem and phloem we can see in the primary structure. And coming to the secondary thickening, the appearance of the root system in cross section, what we can see likewise or appears like vascular bundle-like zones. Actually, it is not well differentiated as similar to dichotomous stem. Okay, so here what we can see, the fascicular cambium is absent because either the bundle-like formation is taking place, then only there the bundle which show the xylem and phloem, and in between the xylem and phloem there is the fascicular cambium. So normally in root system the fascicular cambium is absent because the vascular elements called xylem and phloem, which is derived in a radial fashion, and the formation of cambial strips from conjunctive tissue, which is taking place during secondary growth. So, at the early stages of root development, we can see the cambium is, or the meristematic tissue or meristematic vascular tissue is derived or differentiated from the pericycle, and the pericyclic cell which act as the cambium, and intermittently some points or certain points of pericyclic cell which are immediately de-differentiated into actively dividing meristematic cells, and which produce only one of the vascular element. Either at one point the pericyclic cell differentiated into meristematic cell, and it acts as the cambium, and which produce only one of the vascular element, either xylem or phloem. So either at one point the pericyclic cell or the cambial cell which derives xylem, only that cell which produce xylem elements, and the next or the neighboring next point in pericyclic zone, the pericyclic cell become actually dividing and become meristematic, and it acts as the vascular tissue which produce the phloem. So xylem and phloem intermittently produced from the pericyclic cell at the early stages of development. And coming to the secondary growth in dipole root, we can see the cambial strips which are formed. That is, in the ground tissue inner to the pericyclic zone, some areas or the intermittent parts of the ground tissue which consists of or developed by parenchymatous cells, and which are become termed as conjunctive tissue only when those ground parenchymatous cells become actively dividing or shows the meristematic property, and subsequently it produces the xylem, that is secondary xylem and secondary phloem in the ground area. So that is why we can see some of the cambial strips, that is actively dividing cells which are formed from the conjunctive tissue, that is the ground parenchymatous tissues. And here the three major levels of activity is related with the secondary growth in dipole roots. And coming to the first is the formation of cambial ring. So, as in dipole stem, what we can see during secondary growth, there is a fascicular and interfascicular cambium unite to form the vascular cambial ring. But while coming to dipole roots, here is at the first or early stages of development and the late secondary stages of development, we know that there is no fascicular or interfascicular cambial union. But what we can see here also in root system, there are the cambial or vascular cambial area is differentiated, and here from the cross section or in the diagrammatic sketch, we can see there is the strip of cambium, that is first formed from the pericytic cell, and during secondary growth in thickness, the conjunctive tissue which become act as the cambial or vascular cambial zone, and both the early formed cambial cells and during late formed, that is again the secondary growth and thickness, the cambial cells which are developed, and again at the stages of secondary growth in development, these strips become united and it seems to be a wavy zone or a wavy ring of cambium. So here the first process formation of cambial ring, and here the conjunctive tissue in between the primary xylem and the primary phloem become meristematic, and the forms the strips of cambium. So broken strips of cambium is developed at the first stage of secondary growth of thickness and roots, and all the conjunctive and pericytic cambial cells fuse to form a continuous wavy ring of vascular cambium. A continuous wavy ring of vascular cambium which is developed in dipole root during secondary growth in thickness. So it is the first part of the formation of cambial ring during secondary growth in dipole root. Okay, coming to the second process, activity of cambial ring. So such a wavy continuous ring of vascular cambium is formed. Thereafter what happens? Normally the activity of cambial ring which produces the formation of medullary rays and secondary xylem and the secondary phloem which are produced due to the activity of the cambial ring. And similarly, the secondary xylem which is differentiated or all the derivatives of secondary xylem which is developed during this secondary thickening, and the secondary xylem possesses vessels and parenchyma cells. And here we can see, in addition to the vessels and parenchyma cells, a limited number of tracheids or sclerenchyma cells also developed inside the vascular zone, that is during secondary thickening cycle. Okay. And coming to the secondary phloem, it is differentiated or the derivatives which are developed during secondary thickening. From the phloem are stem tubes, epithelial cells, and also the phloem parenchyma. So here these are the differentiation or derivatives developed during secondary growth and thickness in dipole shoot. And coming to the third process, here we can see the formation of pore or cambium and peridium formation. So for cambium and peridium formation, and here and there in roots there are the lenticels may also develop. It is the third process of formation of both cambium and peridium and also the lenticels during secondary growth in dipole root. And here we can see in this process the core cambium and the secondary cortex is derived in the extrastellar zone or outer to the or peripheral region of the root system in dipole root. So majorly in secondary growth in dipole root, it is consists of three processes. One is formation of cambial ring, and second is activity of cambium ring, and third is formation of both cambium peridium and the lens cells. So this is the process of secondary growth or secondary thickening in dicot root in plants. Okay, thank you.